despite their perennial public health threat that they represent, we have no approved treatments for these. But in response to this ongoing threat to health, we have identified some novel small molecule inhibitors based off of one initial hit in high throughput screening that we think have broad spectrum activity against all the medically relevant enteroviruses. This discovery comes as a collaboration between members of my laboratory group and those of Mike Jung. I'm a pediatric infectious disease specialist, a professor of pediatrics and pharmacology, and I have over 30 years experience as a laboratory virologist, a clinical specialist in infectious disease, and as an investigator in clinical trials of antiviral, antifungal, other antimicrobial agents. Whereas Mike Jung is a synthetic chemist whose experience and successes in medicinal chemistry have included development of a number of antimicrobial agents, and in the oncology realm, molecules that include the successful drug Xtandi that is used to treat prostate cancer. Now, coming back to the scope of the problem then, we're after this because enteroviruses can be mild, but on the other hand, they produce a wide array of readily recognized clinical diseases. The central nervous system infections are probably the most noteworthy because they're the leading cause in the US of viral meningitis. 75,000 cases have been, is the estimate from CDC, and a very conservative guess looking around the world is more than 500,000. But they also cause, as I said, encephalitis, and most recently in the press, which you've seen reports over, how enterovirus D68 has been linked to causing paralysis, very much like polio, sepsis in newborns, hand, foot, and mouth disease, which sounds like a benign disease but isn't. In outbreaks throughout the world has been associated with encephalitis and severe complications as well. And to give you an idea how quickly these come on, I'll just mention a case that I saw here when UCLA's hospital was across the street about 10 years ago, in which a previously healthy newborn did well for a day or so, and then developed fever and irritability, always a warning sign in a small child and a newborn infant. Cardiac arrhythmia developed by day 5 of life. The child was moved to an intensive care unit where chest x-ray and other tests revealed an enlarged and very dysfunctional heart. Liver dysfunction and kidney failure developed, and ultimately this child, despite our best efforts in our cardiothoracic ICU, died at 4 weeks of age from heart failure. This allows me to also mention that these have been seen as a trigger to cardiomyopathy leading to heart transplantation as well. So these are indeed very dangerous viruses that haven't been touched by the fact that we've developed some very potent antivirals. The success in antiviral realm, however, has really been targeted at herpes viruses and, of course, HIV, where there have been tremendous progress. None of those agents has any effect whatsoever on enteroviruses. And in fact, there's really no competitive landscape here. My laboratory was the first group to demonstrate that the antidepressant fluoxetine, in fact, had some activity against an array of viruses, and we hope that that might lead somewhere, but clinical observations and some additional lab work showed it simply doesn't have sufficient potency, even though it has breadth. There is a clinical study in action in which, out there in which an inhaled antiviral is being studied as an inhibitor of one specific type of enterovirus. But even if that works, it doesn't address the fact that almost all enteroviruses are acquired by the enteric tract, by swallowing rather than inhaling, and the disease is far more than just lung disease. And then, of course, you have the idea of using a vaccine, but the vaccine pipeline is its own, is a different can of worms, its own development problems, and vaccines tend to be very narrowly focused. So, in short, we do think that there's tremendous need for an effective antiviral agent. Fortunately, the molecule that we've discovered has some fairly broad activity where the target seems to be a protein known as the 2C protein, crystal structure for the polio 2C, being shown here in this figure. And this molecule in enteroviruses is so conserved that we estimate from our initial testing in vitro that our drug can target more than 50%, our molecule can target more than 50% of enteroviruses. It has low toxicity in preliminary testing in vitro and in our initial exposure in mouse models. And we think viruses are likely to not develop resistance in the short courses that would be needed in order to treat enteroviral infections. And we have this evidence that we are looking at something that could have broad spectrum activity against enteroviruses. But it may not stop there because the structural motifs involved in this 2C protein are found in non-enterovirus as well. So we could inadvertently have found something that has activity against another agent, which I'll mention down the road. How did we get here? We started by doing high-throughput screening of more than nearly 100,000 compounds at the California Nanosciences Institute, using an assay that used live virus, used a pathogenic variant that we knew was very pathogenic in mice and in non-human primates, a Tulane Primate Center, and found that we could use that as a screening assay. Viruses that showed inhibition, protection of cells against virus-induced death, were subjected to a second assay in which a fluorescent virus, we look for inhibition of fluorescence, and then followed that with a quantitative suppression assay, because in virology it's not enough to have some inhibition. You need, because of the exponential ways in which viruses grow, to be able to really potently suppress the number of progeny that you're producing with each replicative cycle. Along the way, we found 21 pyrazolopyridine carboxamides, PPCs, out of a total of about 140 in our libraries. Mike Jung was able to look at this, do some initial structure-activity relationship analysis, produce some more derivatives, which we've exposed to a larger panel of enteroviruses. We've gone as far as looking at the solubility problems for these molecules and been able to get them into solution well enough to do some initial pharmacokinetic analysis in mice. To show you what some of that data looks like, here's a result of a panel of 15 enteroviruses against fluoxetine, the drug that we thought might be repurposed usefully. And suppression is seen. We're using a color scale here where up to 10 to the 6th million-fold suppression of progeny virions is dark blue, and no activity is dark red, and you'll see that fluoxetine seemed to look okay, but it didn't pan out in vivo. There's no evidence it would work. Mefloquine, the anti-malarial, we thought, again, could be repurposed, a drug we could repurpose, but it really looked terrible in our additional screening, and we dropped that. But what we did find instead was that testing, for example, these 4 PPCs, we see good activity against a panel of 13 non-polio enteroviruses, and in addition, PPC number 11 had activity against polio strains, producing a suppression more than 1,000-fold in replication. So that's real antiviral potency. We renamed compound 11JX, and Mike went on to develop some additional derivatives, developing a synthetic pathway for this. In parallel, my lab is working on trying to understand the mechanism of action. And based on understanding the enteroviral life cycle, enteroviruses are really simple. Protein coat, one molecule of RNA, that's it. Once they get in, they release that viral molecule, and since it's message sense, you immediately start to get translation of proteins so you can make new progeny virions. But you need RNA to put in them, and this is the trick. You have to take that RNA, copy it into what's referred to as a negative strand, and then use that as a template for additional RNA synthesis. That's where 2C comes in. It looks structurally related to helicases that unwind nucleic acids from each other. And it's essential. Its activity is essential for replication, and that's what the PPCs do. They appear to suppress the activity of 2C, shutting off viral replication very potently. Now, as I said, we were working with this central pharmacophore. Mike made a bunch of derivatives, and JX17, for example, had much lower cytotoxicity in culture in a variety of cell lines, and it also had higher activity, several-fold lower concentrations needed to get effective suppression in that panel that I showed you. So we took that one, found ways to solubilize it with a variety of excipients and put it into mice, and did some initial PK testing, finding that we were near our, the peak here with JX17, for example, is 3-fold higher than the concentration needed to suppress our usual test virus. And what you see then is, first of all, that we didn't kill mice. Our initial experiences showed us there wasn't immediate toxicity, and we've seen that we can get acceptable pharmacokinetics with really a fairly initial analysis of the problem. Now, I've emphasized the potential to go after the most enteroviral infections that would put you in a hospital. The most recognizable of those is viral meningitis, stiff neck, headache, fever. An easy diagnosis to make, and that is probably a good place to start in terms of trying to clinically test an antiviral agent against enterovirus infections. We could, of course, expand that market subsequently by looking at all severe enterovirus infections and perhaps really any symptomatic enterovirus. I think that's too broad because now you'd be using the virus for trivial things, and now you're starting to potentially get yourself in trouble in terms of of prescribing it and seeing no effect, helping select for community-wide resistance. But nonetheless, symptomatic and life-threatening disease, a good target. But we could go further than that because, as I mentioned, enteroviruses are related to some other very common medically relevant viruses, including the rhinoviruses, which are so similar they've actually been placed in the same genus overall. I wouldn't suggest going after rhinovirus colds, but rhinoviruses are the leading trigger of exacerbations of lung disease in people with chronic obstructive pulmonary disease, in asthma, and in other conditions like cystic fibrosis. But asthma and COPD exacerbations, the rhinoviruses are so well identified as a trigger that, that if we find some activity against them with their similar 2C protein, I think that could be, we could have big impact there too as well. Looking forward in development, we don't see any hard stops ahead, just on the basis of technical challenges. There are ways, we've already found a way around what we conceived might be a difficulty, the limitation to solubility, by finding we can enterally administer this compound to animals, and we have mouse models, and as I said, I helped develop with the Chilean Primate Center a model that we used and published demonstrating that we have viral strains that produce disease in all these species. And then as far as going into a clinical trial for meningitis, for example, this could have been an impediment. It was an impediment in one failed trial that started about a dozen years ago. But now there are FDA-approved platforms so that you can do point-of-care testing and rapid testing in most large medical center laboratories to rapidly diagnose enterovirus infections. So there's a pathway forward to enrolling people in clinical trials. We also have taken great strides to secure our intellectual property, including mentioning as part of our patent applications that the breadth of these is as yet unknown, although we focused on development with enteroviruses in mind. And we see a pathway taking us forward over a period of 15 months from where we are to being able to do challenge experiments, and at least in the murine model, to demonstrate, to look for the best of our derivatives in the PPC class that would suppress replication and that's very easily quantified in target tissues based on our published experience. So I'll be happy to take any questions you have at this time, but I hope I've convinced you we've made good progress towards this goal of addressing this problem that occurs year after year that CDC is now chasing after because of conditions like enterovirus D68-induced exacerbations of asthma and acute flaccid myelitis, a problem that has no solutions yet. I think we've made a good start on looking for one. Thank you.